Good day, everyone. My name is Jin Huang. Today, I'll be presenting this topic, mucosal immunity and translational medicine, to you. First, please allow me to briefing to you about my research background. I did several years of research in molecular biology in academia sinica after receiving my toxicology master's degree from NTU. After that, I went to Johns Hopkins on technology incubation scholarship study pharmacology and molecular sciences because my career goal is to develop therapeutic strategies for unmet medical needs. Since 2017, I went back to academia sinica and joined Professor Edmund Xie's innate immunity research team in GRC. I believe that in-depth understanding of immunology and inflammatory disorders can facilitate development of therapeutic strategies for many inflammation-mediated diseases. Today, my presentation topic is mucosal immunity and translational medicine. I will break this presentation into two parts. The first part is introduction, which meant to guide you through the background of mucosal immunity, food allergy, and type 2 inflammation so that you can better appreciate the contribution of this selected article. And the second part is article presentation. This selected article is from 2019 Immunity, using single-cell RNA-seq to dissect the transcriptome profile of immune cells during type 2 food allergy, by which they discover neuropeptide alpha CGRP controls the expansion of ILC2, and oral administration of alpha CGRP can suppress type 2 food allergy features such as the increase of mast cells and ILC2 cells in intestinal mucosa. Such result implies a potential therapeutic strategy for food allergy which remains to be an unmet medical need, affecting roughly 3% of world population. Part 1 Introduction. Transcriptional Atlas of Intestinal Immune Cells reveals that neuropeptide alpha CGRP modulates group 2 innate lymphoid cell responses. This selected article is the collaborative work of several research institutes in Boston, USA, and Hangzhou, China, Westlake University. Let's quickly look through to get a flavor of the translational medicine and collaborative work. The USA institutes include Broad Institute, MIT and Harvard, Evergrande Center for Immunologic Diseases, Harvard Medical School, and Brigham and Women's Hospital. Mucosal Immunity and Biology Research Center, Massachusetts General Hospital, Harvard Medical School, Department of Molecular Biology, Massachusetts General Hospital, Harvard Medical School, Gastrointestinal Unit and Center for the Study of Inflammatory Bowel Disease, Massachusetts General Hospital, Harvard Medical School, Center for Microbiome Informatics and Therapeutics, MIT, HHMI and Koch Institute for Integrative Cancer Research, MIT, Center for Computational and Integrative Biology, MGH, Harvard Medical School. So this article is working within the context of type 2 food allergy and use a mouse food allergy model throughout the article. Let's first have some introduction about food allergy, its severity and prevalence. You may wonder, why should we study intestinal immune cell profile during food allergy? This is because food allergy is triggered by allergen in contact with intestinal immune cell. Let's first have a look at what FDA said about food allergy. There are these 8 major types of food that elicit food allergy response: milk, eggs, fish, crustacean, shellfish, tree nuts, peanuts, wheat, and soybeans. Here are the known symptoms of food allergy from mild to most severe anaphylactic shock. There are hives, flushed skin or rash, tingling or itchy sensation in the mouth, face, tongue, lips, swelling, vomiting, diarrhea, abdominal cramps, coughing, wheezing, dizziness, lightheadedness, swelling of the throat and vocal cord, difficulty breathing, and loss of consciousness. Severe food allergy could be lethal due to allergic reaction called anaphylaxis. The most severe type of food allergy can elicit anaphylaxis, which is a life-threatening symptom. Anaphylaxis is characterized by two major features: difficulty in breathing and severe lowering of systolic blood pressure. Persistent GI tract symptom is another main feature. The common scenario of the onset of anaphylaxis is exposure to allergen through skin or mucosal tissue. And food allergy remains to be an unmet medical need affecting about 3% of world population. Currently, there are some therapies for food allergy as listed above: oral immunotherapy, sublingual immunotherapy, and epicutaneous immunotherapy. However, the most effective ones, oral immunotherapy, also have major side effects such as anaphylaxis. And all these are saying that we need a better therapeutic strategy to manage food allergy. When it comes to the significance of this article, briefly, there are three major significant points made by this article. Firstly, the authors are the first ones to systematically characterize intestinal mucosal immune cells. Secondly, they discovered that alpha-CGRP involves in modulating ILC2 responses, including ILC2 expansion and cytokine secretion. Lastly, their novel finding has the potential to be applied in clinic treating type 2 allergic patients. The points listed in the right-hand side, I'll go through these to guide you through the major background knowledge and concept of this article so that it would be much easier for you to appreciate the contribution of this work. Let's first look at the anatomy of intestinal mucosal immunity. First, let's look at the upper right photo. These bulges are Peyer's patches. They are structured secondary lymphoid organs. Lamina propria are the loose structures surrounding Peyer's patches. Although not so well structured, still enriched of many immune cells. The reason to introduce these two structures is because they are the main region where immune cells locate, and later studies in this article dissects the atlas of immune cell transcriptional profiles from both lamina propria and Peyer's patch. Food allergy animal model used in this study. The OVA-induced food allergy mouse model. To simulate human food allergy, the mouse food allergy model is established as such. Firstly, the mouse are sensitized with allergen, here OVA. OVA stands for ovalbumin, which is a protein with 385 amino acids, and its molecular weight is about 47.5 kilodalton. Through intraperitoneal injection of 50 microgram of OVA and 1 milligram of adjuvant aluminum potassium sulfate twice, 2 weeks apart. After another 2 weeks, oral challenge of 50 microgram of OVA for 5 times every other day. After 5 times of oral challenge following sensitization, the mouse is now considered to have developed type 2 allergy and is then subjected to further studies. The intestinal mucosa immune tissue, LP and PP, were then taken from these animals for further analysis. So, what is known about type 2 allergy? What are the known knowledges? Please let's look at the following slides. Type 2 inflammation, the knowns. Let me briefly remind you the differences between type 1 and type 2 immunity. Type 1 and type 2 are separated based on the type of T helper cells that are involved. Type 1 immunity has T helper 1 cells secreting cytokines such as IL-2, interferon gamma, lymphotoxin A, and is characterized by prominent phagocytic activity, whereas type 2 immunity has T helper 2 cell secreting cytokines including IL-4, IL-5, and IL-13, and is characterized by high antibody titer and eosinophilia. In 2015, Nature Reviews Immunology has summarized all cellular and cytokine components involving in type 2 inflammation. The left-hand side summarizes are the complete list of all involving factors. Notably, there are many cell types involving besides ILC2. Now you might wonder why the author later only focused on addressing ILC2. I can tell you now that REC1 deficient mice, which does not have any mature T or B cell, still develops type 2 inflammation. On the contrary, ILC2 deficient mice showed significantly reduction of type 2 inflammation features in terms of cytokine levels of IL-4, IL-5, and IL-13. ILC2 and type 2 immunity. ILC2 develops from common lymphoid progenitors but lack the surface expression of the common lineage markers, and its function mirrors T helper cells. ILC2 produce Th2 cytokines regulated by GATA3. GATA3 is the master Th2 cytokine transcription factor, which is highly expressed in CD4 Th2 cells and has been shown to be critical to the development of and function of ILC2. In 2014 Immunity article, Group 2 innate lymphoid cells are critical for the initiation of adaptive T helper 2 cell-mediated allergic lung inflammation. In this article, the author has shown that ILC2 deficiency severely impair allergic inflammation, and then they use a bone marrow transplantation model to demonstrate that the relevance of ILC2 in type 2 cytokine abundance and serum IgE level, because bone marrow of RR alpha knockout mice is deficient in ILC2, and in response to allergen stimulation, the lack of ILC2 corresponds to prominent decrease of type 2 cytokine, as well as serum IgE, as well as lung GATA3 positive cells, and this implies that ILC2 works upstream to plasma cell and mast cell in type 2 inflammation, and in lung, most GATA3 positive cells are infiltrated ILC2. All these evidence support that ILC2 plays a critical role in initiating type 2 immunity. Type 2 inflammation, IL-25, ILC2, IL-5 axis. When chemosensitive cells, tough cells, in the epithelium lining of the gut comes into contact with pathogens or allergens, it secretes IL-25, which then binds to the IL-17 receptor beta on ILC2 surface membrane to elicit its production of IL-13 and IL-5, the major cytokines brings forward type 2 inflammation. IL-25 is highlighted here because it is later used in this article to mimic the launch of type 2 inflammation, notably IL-13, IL-5, mast cell number, and ILC2 number are all used as endpoints measuring type 2 response in this article. Many cell types are involved in type 2 inflammation, and this slide shows the lineage tree of myeloid cells and lymphoid cells. Pink highlights the cell types that are known to play a role in type 2 inflammation. These known knowledge are later systematically examined in this article.